Thank you once more for joining us today for the Potato Late Blight webinar, Unlocking Microbial Solutions for Farmers. Today, we have a fantastic event, which is packed with some excellent information for you today and some fantastic panelists. So to start off, I will walk you through some housekeeping before we kickstart the session today. Please use the chat function to ask questions throughout the webinar session, and we will make sure to get through all of your questions at the end of the Q&A session that we have scheduled in. Should you require any tech support, please reach out to our colleague, Nicole Hutton, via the chat function. And do mind that the session today is recorded. The recording will be made available via YouTube, and we will make sure to provide you with the link. For today's program, we will start the session with a welcome and an introduction by CHAP's very own Richard Glass. Afterwards, Mark Wilkinson from Bactobio will be providing us with a company overview, followed by Carrie and Maisey from Bactobio. They'll be walking us through scientific overview of microbiome work. And afterwards, David Cooke from the James Hutton Institute will be talking about population change, fungicide efficacy, and IPM. And last but not least, CHAP's very own Alex McCormack will be walking us through the wider influences and agri-tech projects. And last but not least, we will be having an engaging and fantastic session of Q&A. And now, without further ado, I would like to pass on to Richard Glass from CHAP. Richard, the virtual stage is yours.
Thank you very much, Andrea. Good morning, or good afternoon, rather, everybody. So I'm just going to give a brief introduction to this project that we've been doing with Bactobio. So it's an Innovate UK project funded by DEFRA, 18 months long. So we've been doing work at Rothamsted Research for some implanter studies, as well as doing some field work to do soil sampling, taking samples of soil and rhizome before blight gets established in the potato field, and then at the end of the season when we have more blight in the field, and looking at the change in the microbiome over that period of time. So the samples that we were taking early and late season were sent off to Bactobio, and they've been doing the extraction and further analysis of the microbes in those samples. So I shall hand over to Mark Wilkinson, who will present details of what they've been up to. And just so Mark is the co-founder and chief scientific officer at Bactobio, a biotech company based in London focusing on finding new antimicrobials from previously uncultured microbes. Prior to Bactobio, Mark completed a PhD in molecular parasitology and worked as an innovation fellow at GlaxoSmithKline. Mark founded Bactobio 3.5 years ago, and since then the company's attracted around £14 million worth of funding and grown a team of 45 scientists. Mark will provide an overview of the company and more details about our joint project. Over to you, Mark.
Thank you very much, Richard. Can you see my slides?
Yep.
Perfect. So thanks very much, Richard. Yeah, I'm Mark Wilkinson. I'm Chief Scientific Officer and co-founder here at Bactobio. And today I'll be giving you a brief overview about the company and our technology, and then going into a bit more detail about this project that Richard has briefly introduced there. And then I'll be handing over to two of our scientists to go through a bit more of the detail on the microbiome analysis, and then also how we're using machine learning to find new bio-derived fungicides. So Bactobio is a London-based biotechnology company that's about 3 years old now. And the company was founded on harvesting valuable metabolites from nature and turning them into antibiotics and fungicides. So microbes represent nature's greatest bioresource. When we think about phenotypic differences, so changes in genetics, we might think about elephants with tusks to defend themselves, or birds with wings to fly away. But actually, the the genetic diversity in these multicellular organisms is very limited compared to that of microbes. So if we look at this tree of life on the screen here, what we can see is microbes span a huge genetic space. And this genetic space corresponds to a chemical space which is equally as diverse. So these microbes produce these compounds to kill one another. So if a bacteria kills another bacteria, that compound can be repurposed as an antibiotic. And so actually around 80% of all antibiotics that you'll take today derive from soil microbes. Similarly, if these microbes kill other fungal pathogens, they can be repurposed as fungicides. And that's exactly what we're trying to do with this project. And that combined, microbes have captured markets worldwide of around $250 billion. But what's really interesting is actually these solutions have derived from just 1% of microbes that can be readily cultured in the lab. So in order to find these compounds, we need to bring the microbes into the lab, grow them in isolation, and then isolate the compounds that they produce. But the remaining 99% of microbes have been completely unexplored. And so these species often grow together and rely on one another for specific food materials to grow or specific growth conditions. But when you take a sample, so if you take, for example, a sample of soil, there'll be around 10,000 different microbes in that soil sample, but only 100 of them will readily grow in the lab. And so at Bactobio, what we've done is built a platform that provides access to this remaining 99% of previously uncultured microbes. So our platform broadly has 3 steps. The first is microbial generation, so the discovery of new microbes. The second is compound discovery, so looking at the compounds that these microbes produce by screening them against specific pathogens. And then the third step is commercialization and further development of these compounds to ultimately deliver products to market. And at the company, we've built a library of around 2,800 microbial species. And so for context, our library has around 5% of bacteria that have ever been cultured worldwide. And what you can see from this slide is the microbes are all kind of weird and wonderful shapes and sizes. And what you see is that a lot of these microbes are producing different colored pigments here. The reason they produce these pigments is because the genetic information they contain codes the compounds to produce these colored elements. But we're interested not in the colored elements, but the compounds that kill other pathogens. And so when we started this project about 1.5 years ago, we'd already validated our platform for discovery of new antibiotics, and we aimed to validate the transferability of our platform from antibiotics to fungicides. So how do we go about this? So this project, as Richard said, is an 18-month project that had the main objectives of validating our transferability of our platform to discover compounds that were active against potato-like blight or Phytophthora infestans, and then isolate these compounds so that we can develop fungicides. The methods we used were to use our platform for new bacterial diversity, isolate these compounds, and then ultimately test them in Panther, providing project outputs as soil health data from across farms in the UK and new bio-derived fungicide compounds. So broadly, the project had a number of different technical areas. And the first, as Richard said, was understanding the microbiomes across these soil farms. So the reason we do this is to potentially identify microbes that were able to fight the pathogen that causes potato blight. We then identify these microbes and understand the genetic material behind the compounds that they produce, screen them for activity against Phytophthora, and then test them in Planter. So the first step of this is soil sampling and microbiome analysis. So as Richard said, a few of the team members from CHAP, Richard, Sophie, and Archita went out and collected soil samples from before and after a Phytophthora infection. And what Carrie is going to talk you through today is how we use this information to potentially identify microbes that are associated with defense against the disease. The second step is to further characterize these microbes and understand the genetic information, isolate them in the lab and understand their genetic information to produce these compounds so that we can prioritize the microbes that we're screening against the pathogen. In this project, we also developed a high throughput screening assay against Phytophthora. And so on the left-hand side, what you can see is the traditional approach for the discovery of new compounds, where you have your fungal pathogen that grows around the outside, you're producing isolated microbe in the middle, and then a clearance zone where the compounds produced by this bacteria are defending against the pathogen. Now, this has been very efficient, and if we go back to kind of the early 20th century, this is the method by which penicillin was discovered. However, at Bactobio, we aim to routinely discover new compounds. And to do that, throughout this project, we've developed a high throughput screening assay that allows us to go from 1 bacteria per experiment to over 384 per experiment. And this really enables us to do high throughput screening to discover these new compounds. And then ultimately, as we grow our compounds up in our bioreactors that we have on site, we can then purify out the active substances and then test them in planter. And so today, we've got our 2 scientists. We have Dr. Carrie Alderley, who's been at the company for around 2.5 years now, and has a PhD in microbiology. And she's a microbiologist at the company who uses genome sequencing and genetic sequencing to understand what microbes do. And then we have Maisey, who's a scientist who's been at the company for a similar amount of time, who's a computational biologist. And Maisey is building the machine learning tools that we need to identify the genetic sequences associated with compound production and fungicide production. And so I'm now going to hand over to Carrie, who's going to give an overview about the microbiome work, and then she'll hand over to Maisey, who'll go into our machine learning.
Thanks, Mark. Hello. So I'm going to be talking about microbiome analysis across farmland. So you may have noticed that within one potato field, certain potato plants will show differences in the levels of severity of late blight disease that they show. So some potato plants will show severe symptoms of late blight disease, while others will show no symptoms at all or very mild symptoms. These differences in disease progression rates can be partly explained by what's going on underneath the soil surface with the millions of soil microbes that coexist in the soil there. This work package aimed to identify these bacteria that exist in the soil that are able to produce antimicrobials that are effective against the late blight disease pathogen. So we aim to answer 2 questions. Firstly, how does infection affect the microbiome composition? And secondly, what kinds of bacteria are associated with defense against late blight disease and are responsible for establishing these disease suppressive soils? So to do this, we received soil samples from healthy and diseased potato plants, and we extracted the DNA from these soil samples and sequenced the DNA. So if you imagine at the supermarket, every item of shopping that you buy has its own unique barcode that can be scanned to identify what that item is that you're buying. In a similar way, every bacterium has a unique part of the DNA code that can be sequenced or scanned to identify what that bacteria is and what group it belongs to. So we aim to do this DNA sequencing from these soil samples to build up a picture of which bacteria are responsible for creating disease suppressive soils. We received soil samples from a farm in Aberystwyth in South Wales, and soil samples were taken at 2 sampling dates, one in early August, a few weeks following potato plant germination, when disease symptoms were beginning to become evident, and then again at late August, when disease symptoms were widespread across the field. Potato health symptoms were assessed based on leaf disease symptom severity. So here you can see what we'd consider a healthy potato plant versus a diseased potato plant. And soils were sampled from the bulk microbiome from the base of the potato plants and from the root zone. Some of the potato plants were treated with fungicide, Infinito, whereas others were untreated. So plant health status was determined at the early sampling date, and half of the samples were healthy, from healthy potato plants, and the other half were from diseased potato plants. However, by the second time point, a large proportion of the plants that had originally been assessed to be healthy had developed disease symptoms. And so we wanted to focus in particular on these potato plant samples that remained healthy and might have a disease suppressive microbiome that enabled them to evade infection. So one measure of soil health that we were particularly interested in was microbial diversity. And this is a measure of the number of different bacterial groups existing within a soil sample. So if you imagine here, we've got 3 different microbial communities, and each colour represents a different bacterial group. So for instance, community A has 5 different colours, and so 5 different types of bacteria. And similarly, community B has 5 different colours and so has the same diversity score as community A. However, community C has just 2 different colours, yellow and blue, and so we'd say that has a lower diversity than communities A and B and only has 2 different soil bacterial groups associated with it. Diversity is really important as it's an indicator of soil health, and soils that are made up of many different bacterial groups and have a higher diversity are associated with increased disease resistance, faster plant growth, and higher crop yields. When we investigated the diversity of microbes within the soil samples we received, in the bulk root zone at the early sampling date and at the late sampling date, we noticed consistently that samples from diseased potato plants had reduced diversity than those with healthy. So if we refer back to the cartoon that I showed earlier, you can imagine that these diseased potato plants had fewer bacterial groups associated with them than the healthy ones. And this has implications for the future for secondary infections as well. When we looked in more detail about the bacterial groups that were affected by disease and those that were associated with soil samples that with potato plants that stayed healthy from the first time point to the second time point, we found that there were 6 different bacterial groups that were enriched in these healthy samples and 2 that were related to disease samples. We wanted to focus in particular on these 6 bacterial groups to give us a better direction of which bacteria might be responsible for releasing antimicrobials that can target the Phytophthora infestans pathogen and who might be useful to us to identify new bio-derived fungicides. We also looked at the effects of fungicide treatment with the synthetic fungicide Infinito on the potato plants. And we found that, as expected, those potato plants that were treated with the fungicide had reduced disease symptom severity than those that were untreated. However, when we looked at the sequence data, we found that those that were treated with the fungicide had lower microbial diversity than those that were untreated. And this is particularly concerning as it may have implications for future disease development and soil health. Of even greater concern, when we looked at which bacterial groups were affected by fungicide application, we found that 2 of the bacterial groups that were associated with healthy plants and potentially responsible for disease suppressive soils were suppressed by fungicide application. And this highlights the importance to find new bio-derived fungicides, which are less likely to have detrimental effects on the soil microbiome and these bacteria that are important for disease resistance. Now I'm going to pass over to Maisey Davidson. She is a computational biologist at Bactobio, and she's going to be talking more about how we can use machine learning to identify new fungicides.
Cool. Hi, everyone. Thanks, Carrie. So I'm going to tell you how we then find these fungicides from the bacteria. So as Carrie and Mark explained, the soil around the potato is a microbiome full of bacteria. And as Carrie showed, different bacteria here can help protect potatoes from blight disease. And the way that they can actually stop the disease from developing is by producing fungicides. So in nature, these fungicides are naturally produced to help the bacteria survive. It helps reduce competition and helps them gain more nutrients. And so if we can isolate these compounds, we can use them ourselves by directly applying them to potatoes to help protect crops. And so to understand how to do this, we have to think about what are these fungicides and how do bacteria make them. So fungicides are just complex chemical compounds that can kill fungus. And so an example here on the left is nystatin, which is a fungicide produced naturally by bacteria. And you can see that it's quite a big complex compound. For comparison, the one on the right is glucose, which is just sugar. So bacteria make these compounds using a production line of enzymes, and they build up small compounds step by step until they get the final compound. And so you can think of this as like a production line in a factory. The compound goes along the conveyor belt and each enzyme adds a different component to the chemical compound until you get the final product. And so the instructions to do this are contained within the bacteria's DNA. So this is like the manual for producing these compounds. Each gene in the DNA produces one of the enzymes in this production line. And these genes are all grouped together in the DNA, and we call these groups of genes biosynthetic gene clusters. And so since this is what controls the production of the compound, we're really interested in understanding these because it can help us predict which bacteria will produce compounds and what kind of compounds they'll produce. And so we try and apply this to our pipeline. So as Mark explained earlier, we isolate novel bacteria from the soil, and then we bring them into the lab so that we can grow them. Once they're in the lab, we can extract their DNA and sequence them. And so this is slightly different to the DNA sequencing that Carrie talked about. Because we've grown them individually in the lab, we can sequence their whole genome rather than just that barcode. And this gives us a lot more information. From these whole genome sequences, we then can run machine learning software that predicts and finds these biosynthetic gene clusters in the DNA. So the software looks for these genes. It looks for genes that it knows can make compounds, and it looks for groups of them together, because when they're together, that's when that production line can happen. At Bactobio, we have over 1,000 genomes, and in a single bacteria, it could have anywhere between 0 and 50 gene clusters. That means in our collection, we've got over 10,000 gene clusters. And so we need a way of prioritizing them and making sense of these gene clusters. One of the ways that we do this is by comparing them to databases of known gene clusters that produce fungicides. So these are gene clusters that other people have studied before, and we know what compounds they make. So when we compare our gene clusters to public gene clusters, we can find matches and similarities between them. So whether the genes are the same, whether they're in the same order. And when we do this, there are a few different outcomes. So the first is we might find clusters that are very similar to known gene clusters. All of the genes are the same, and this means that they would produce the same compound. So for example, if a cluster matched one that produced nystatin, the compound that I mentioned earlier, we would think that this cluster would also produce nystatin. And so we would not be so interested in these because it will just be producing compounds that are already known. The second kind of cluster we could find are some that shows some similarity to known gene clusters. So the first gene's different, but the rest of them are the same. And so we would expect these to produce new but similar compounds. And so they might have a slight edge, they might be able to overcome some resistance, or they might be more potent, but they'd be acting in the same way against the fungus. So they'd have the same mode of action. And so this is still useful, but the final category is the one that we're really interested in. So when we find clusters that are not similar to any known gene clusters, these are the ones that are likely to have a new mode of action and be a completely new compound. And so these compounds are important because they the pathogens won't have resistance to them yet necessarily. And so these are the ones we're interested in. Because we don't know as much about them, we still have to test them because, as Mark showed earlier, sometimes these bacteria can just produce colored compounds that are not that useful, but some of them might be the fungicides that we're interested in. So once we've prioritized the bacteria that we're interested in, that's when we take it through for testing. So we carry out the high throughput screening that Mark described earlier to identify the bacteria that are producing these active compounds and validate the earlier work. And once we found that, we need to grow up these compounds in much larger quantities. So we do that by growing bacteria up in these big flats like this, and that allows us to isolate much larger quantities of the compound. Once we've done that, we can take the compound into the plant trials. So this is currently underway, and we're testing some of our compounds now in glasshouse trials. We're interested both in whether the compound is able to protect the plants from becoming infected in the first place, and whether they're able to eradicate infection that's already started. So in the pictures here, you can see how compounds are applied to the leaves before infection with P infestans to see how well they work. And so we're currently eagerly awaiting these plant trial results. So to sum up what we've done in this project so far, we've been using microbiome analysis to guide our search. We carry out genetic analysis to identify which bacteria we think will produce the most promising compounds. We then test these bacteria for activity, and we purify the compound. And then finally, we test the compound in plant trials and take it for further development. Crops are increasingly becoming resistant to our current fungicides, and so it's really important that we find new compounds to help with food security and protecting our crops. And so ultimately, the aim of this project is not just to find one compound, but to build up our pipeline so that we can consistently find these new fungicide compounds. Cool, I'm gonna pass on to Richard next. Thank you.
Thank you very much, Maisey. And now we have David Cooke to give his presentation. He is a graduate of the University of Wales in Aberystwyth, where he studied agricultural botany. He's worked for the former SCRI, now James Hutton Institute in Dundee, for almost 30 years as a plant pathologist, studying the detection and evolution of Phytophthora. He's characterizing populations of Phytophthora infestans and how this impacts late blight IPM on a UK and European scale. Since 2003, we're working within a multidisciplinary group at the Hutton Institute. David, over to you.
Okay, thank you very much for the introduction, Richard. Can you hear me and see my slides okay?
Yep.
Okay, that's always a good start. Excellent. Yeah, okay, so a slightly different tack now. I'm not part of the project, but I was invited to come along and join this very interesting sounding symposium to add some other context and other work on Phytophthora infestans, the pathogen we've just been hearing all about. So firstly, just before I go any further, I'd just like to remind people that Phytophthora infestans, isn't a fungus in the sense that we've heard about introductions to microbial populations. It is rather startlingly, it's an algae, and it's as closely related to fungi as we are to pine trees. So it's always quite good to have that context in mind when thinking about actives and what's going to be active against blight compared to other things. So, but anyway, moving on. So I'm calling in today from Dundee. Myself and Alison Lees run projects here in a group on Phytophthora infestans on a whole range of different things. And I'm going to specifically talk about the Fight Against Blight campaign and the impact of that. But just first, I want to sort of just stress the type of work we're doing up here at Hutton. So it's fundamental biology and applied research, and we're always looking for the impact of that. So one of the first things we're doing is population tracking. So why would we need to understand changing populations of the pathogen? Largely because of the fact that the pathogen evolves, and it evolves in response to things that we do to try and manage it. That'll be host resistance and fungicides are the two key things. And fungicides, of course, are highly effective at controlling late blight. But as we've just heard, they do have impacts, and indeed the pathogen can become resistant to them. So we do quite a lot of work on variety resistance and fungicide sensitivity monitoring, but we also do a lot of fundamental research on pathogen and host biology, really understanding the interaction, how the pathogen interacts with its host. So that includes genetics, genomics, and recently some work on microbiome and biocontrol research. So Lorena's just started with us looking into that area as well. We're very keen on communicating these findings to industry, and the whole point of all this is to support best practice or integrated pest management, so which fits nicely into the subject of the seminar today. And on the right there, you can see a rather nice as a plant pathologist's perspective. Sorry, if you're a grower, that wouldn't be nice. It's a stem lesion of late blight, and then the real problem is when it moves through onto the tuber. Just thinking, I was going to get my pointer going. But anyway, you can see that on the right-hand side there, the tuber blight. Before I go on to the fight against blight monitoring, I thought I'd just flag very briefly the testing we've done on biological efficacy in field trials. This is part of an EcoSol project, and it's very briefly, we've got a susceptible cultivar here in the field with a disease which has moved from 0 in early July, up to 100%. So clearly untreated plot to become heavily diseased. And there's a couple of coded products here that are showing, encouraging levels of efficacy. This bodes well for the project that CHAP have and that Bactobio are working on. We can see that biological products, can have effects in the field. I don't think I'm giving away any state secrets to say that there's been some skepticism, or if you like, or concerns that biologicals won't be as effective in the long term against some of the very effective synthetic chemistry. So there's signs of hope there on this susceptible variety. At the bottom here, we've got other fungicides integrated, the biological fungicides integrated with other lower dose of fungicides and other IPM strategies. However, another point here is that the resistant variety, carousel, the first thing you should notice here is we've got 100% disease here, and we've only got 8% disease with the resistance. So resistance works very well. And even more encouragingly, when you combine the biologicals with host resistance, you end up getting a product which is, doing a good job. If you try to convince somebody, a grower, that 70% disease compared to 100% disease is good, that wouldn't be a very convincing argument. But this on the right-hand side with a resistance variety is. Okay, moving on. FAB stands for Fight Against Blight. So we have sponsorship from 16 different commercial organizations. So I'd like to thank them first before I go on to explain the population change work. And why were we doing this? Excuse me. The context for this particularly was a group of fungicides called CAAs, which are extremely important for blight management. They're the pillar of many blight management strategies across Europe. And with colleagues in Denmark and in the Netherlands, we discovered that there was a strain of blight which could overcome these fungicides, and it's called EU43A1. And as this map here shows, it spread from Denmark through to the Low Countries, and we were very concerned about it spreading to the UK and having a big impact on blight management in the UK. So that was the context. Going on to the 2023 season now, I'll just present results from this season specifically. And in a nutshell, it was a very strange season, hot and dry in May and June. In July, we had some very severe rainfall, as you can see from the rainfall map. And at the bottom, the chart shows the number of samples that came in over this time. So there are a lot of samples arriving, in which we were testing for all sorts of traits. And this graph just shows you, some key statistics, if you like. So 196 outbreaks were sampled. We've got about 1,500 of those samples came in, and 830 of those have been genotyped. The way we tell which blight strains are impacting our crops is to genotype them with some DNA-based microsatellite markers. And also in context for this, through contacts we have with the mainland Europe, we know that severe blight was suffered across the Low Countries of Europe. Okay, so what did we find? Again, in a nutshell, in one slide, which summarizes an awful lot of work, this shows population change over time, and the different colored blocks represent different clones of the potato late blight pathogen. So, I mean, jumping straight forward to 2023, obviously we've got a lot of change. I suppose the first thing to say is you can see the transitions over time. We've had a blue strain which has appeared, which was resistant to a fungicide, a pink strain which was just highly aggressive and doesn't seem to have been impacted by fungicides. And then more recently, this dark green strain called 37, which was resistant to a key fungicide. The sort of salmony pink coloured one is highly aggressive, but we don't think it has any fungicide resistance. And the 43A11, which I just mentioned, the CAA resistant one, well, the good news was it hasn't been found in the UK in 2023. So that's the main take home of here. I suppose the take home of this slide is that the populations can change, and so far we don't have this highly resistant current one. So we've deferred the problem for another year at least. Okay, if we look at that across the UK as a whole, you can see that the population varied according to the region, but I don't think I'm going to go into much detail on that today. I think we're better off keeping some of the information and keeping time for Q&A. So I won't go into the details there. It's just to say that England and Wales, are slightly different from Scotland. The population up here in the north lags, if you like, a little bit from the population down south. You can also look at the details on the euroblight.net website. At Hutton here, we are joint coordinators of the Euroblight system. So I've been genotyping samples and putting the data into the database, and that is all then plotted and freely available on the euroblight.net website. So have a look at that. Okay, last couple of slides. I want to focus now on fungicide efficacy. So really, all those sponsor companies I showed you at the beginning are producing a vast array of different active ingredients. But as part of this project, we wanted to do some independent testing of these fungicides for their efficacy against different strains of potato late blight. So work that we've been doing here, and I'll just show the methods in a very brief way. We're talking about a detached leaf protectant assay, and we inoculate with zoospores, and then in these assays, in this assay system, you can see on this occasion you have isolates which have generated lesions with the fluffy growth around them, and other isolates which you can see there's no lesion on those leaves, so the fungicide has been very effective against those. We've tested a lot of different fungicide combinations with different doses, and we've been calculating the EC50 values. That's a measure of a dose at which kills 50% of the disease, or controls the disease to a level of 50%. Now this slide summarizes an awful lot of information, but you don't need to look at the details. The key thing is the active ingredients, so zoxamide, amatoxogen, amisulbron, propamacarb, and oxythiopiprolin. These are 5 clearly important active ingredients which were selected by our sponsors and scouts throughout the industry. The main point is that we've tested them, and none of these coloured bars, which represent the different clones, have shown any resistance to these 5 products. There is variation in the height of these peaks, but if you relate that and the values you can see on the y-axis to the values in brackets after each of the fungicides, that's the ppm field rate. So although there is variation for oxythiopiprolin, it's so far away from the 75 ppm field rate that it's, they're not biologically significant. So it's in terms of the UK, this testing has been communicated to industry, and it's good news in that these range of actives will be effective. However, on my summary slide, you'll see that not all the news is so positive on a European scale. So coming to some conclusions. Firstly, I hope I've convinced you that knowledge of the population and the evolving population is important, and that will be the case for biological fungicides in the same way as it is for synthetic fungicides, incidentally. But there's just a greater range and potentially new and exciting actives there that we will hopefully discover. The fungicide programs for next year are going to be complicated. Okay, I know not everyone has a detailed knowledge of the fungicides, but the CAAs are the ones I mentioned. before. So resistance is already confirmed, but we haven't found that in the UK. I've already said that. But resistance concerns do continue in mainland Europe. Fluazanam, the one that green clone I explained is resistant to, yeah, it's declined in the UK. And that's good news because that product can still be used because the population has been less fit and has declined in the absence of its use or the reduction of its use. Metalaxyl is an old product, the one that very dark blue clone is resistant to. That's also declined, which means that product is active and available again in the, can be used again for fighting blight in 2024. Oxythiopiprolin is a very good product. For those of you in the industry, you'll know this is called Zorvec and produced by Corteva. And, it's proved to be highly effective, but this year, and it's a bit of a big but, especially if you're living and working and growing potatoes in the Netherlands, resistance has been found. But we don't have any evidence for that in GB samples. But we need to be very careful and plan fungicide use in 2024 to make sure we don't select for resistance in the UK population. And the last couple of points, cultivar resistance. So we know that really varietal resistance is going to be important and increasingly important with biological synthetic fungicides and the whole management of late blight. And also, getting other products and ranges of products as we lose existing ones is also important. Lastly, primary inoculum is also important, key to know about. These pictures here show what happens when you get the sexual inoculum in soils, which germinate and can kill plants early in the season and then spread through. And they generate these, if you have oospores, you have sexual recombination within the pathogen population, which makes the pathogen much more able to adapt, of course, to the selection pressures or the pressures that we apply to it. Okay, and at that I will, it's the end of my presentation. Thank you very much. I'm very happy, along with everyone else, to answer questions. Thank you.
Thank you very much, David. And yes, please do put your questions in the Q&A session, and then we can have a discussion at the end of the presentation. So next, we have my colleague from CHAP, Alex McCormack. He's an innovation sector lead with a strong background in agriculture, spanning work on farms and industry and R&D. He has a PhD from Harper Adams University and has also worked at the University of Warwick, investigating novel disease management in vegetable crops. He's involved with a number of potato projects at CHAP at the moment, and he's going to give you a brief overview of some of his expertise. Over to you, Alex.
Thank you very much, Richard. So very much following on from David, I'd like to kind of use my presentation just to expand the context and the wider landscape of potato production out a little bit further, just to look at the sector changes that we've witnessed, some of the challenges, and also looking very much to the future and how some of the projects that we're developing at CHAP and across the industry can kind of help with that. So to begin with, there are a number of key challenges that face the sector, but from my point of view, I picked out a couple that I'd like to talk about today. Already we've heard about the losses of key active ingredients or pesticides, and how this is a general trend that we're seeing, that there are less pesticides coming through production pipelines, and we're also seeing reduced efficacy and potentially the increase in the occurrence of resistance amongst populations of pests and diseases and weeds. Alongside this, particularly for potatoes, a big elephant in the room is the amount of soil really moved during the cultivation and growing of that crop. And as we transition into a more regenerative agriculture, where we're looking even more to enhance and look after our soil health and structure, potato cultivation can offer some problems to that. Looking much more longer term, we also need to look at how the industry can help develop new integrated systems for potato production, both from a pest, weed and disease management point of view, integrating cultivations with other wider practices like regen, and also looking at the whole production cycle as well, so going into crop storage, which also has its own problems, but also opportunities. So just to give you a bit of a flavour, I've picked out a few of our project case studies. So alongside Bactobio's project, we also have been running a variety of others with the potato sector to try and tackle some of these key challenges. The first one I'd like to talk about is the DCIS project. Here we're trying to use a trap crop to manage the potato cyst nematode, quite a common problem in potatoes across the UK. And this project is just coming to the end of its 2 years. funded by Innovate UK through its Farming Innovation Programme, and is a collaboration between CHAP, Produce Solutions, VCS Potato, Harper Adams University, and a consortium of growers as well. So really this feasibility study came about because the potato cyst nematode, these round cyst structures that you can see on the roots on the left there, is a common problem across the UK. So about 48% of soil in the UK is infected with PCN, these little cysts that you can see. And each one of those cysts might contain up to a couple of hundred eggs of which are able to hatch into small juvenile nematodes, very small microscopic worm-like organisms. They are able to live in the soil for quite a long period of time. So we typically grow potatoes over a rotation of about 1 in 6 years, and PCN are able to survive potentially for up to that long. What they do is they hatch out of these cysts and they infect the roots of the potato, and once they are into the roots, they form feeding sites, which can negatively impact the yield and quality of those potatoes. So you can get misshapen tubers, very small potatoes, not very good for making salad potatoes or crisps or chips and things like that. Currently, the sector manages this problem through a variety of tools. So there's that long rotation, typically 1 in 6, but going longer might be required where we see heavy PCN infections. You can use it through, manage it through other means, but primarily the use of synthetic nematicides is the most common practice. But as with many active ingredients in traditional chemical pesticides, these synthetic nematicides also have an uncertain future, with some of them becoming coming up for their re-registration soon and might not be re-registered. So what we need as a sector is new holistic solutions coming through to try and tackle these challenges. And even if we have synthetic nematicides continue to be used, growers still need more tools in the toolbox to stack around them to use as a whole kit of tools to manage PCN. The solanaceous trap crops, which are branded under the name Decyst, hence the project name, offer one such solution. They are able to elicit the same response as the potato plant. They are solanaceous, they are relatives of the potato, and so they produce root exudates that causes the cysts in the picture there to hatch and move towards their roots. The nematodes think it's a potato, they infect the root, but through a variety of biological mechanisms and also the period of time that the crop is in the ground, they are trapped in the roots and unable to complete their life cycle, thereby kind of removing them from the soil. This is a great idea, and we've had these kind of trap crops available on the market now for 10 or 15 years, but their uptake by growers has been largely quite low due to a variety of agronomic reasons primarily. Great difficulty in knowing how best to sow the crop. It's a very small seeded crop, not dissimilar to sesame seed or tomato. Not knowing how best to manage that crop. In order to get the good root exudates, you need a good, healthy crop, lots of biomass, lots of growth, which can be quite difficult for growers to achieve. And then we also need to know how best to get the most out of these crops. So we need to look at how best to maximize efficacy through the period of time they're in the ground, alongside looking at new types of solanaceous trap crops. So for example, a new species here known as Decyst podium, alongside another one known as broadleaf. We've tested these in efficacy trials. And really the project's aims were to very much put them in the hands of growers and agronomists, see if we can really optimize the management of these trap crops to maximize their efficiency, and then to use these demonstration trials to garner more interest from the sector, alongside producing a variety of video and written materials to help growers understand more and get the best success that they could do with trap crops in the future. So we're now in the final year. We've spent about 2 years of doing this now, and we've looked at a variety of agronomic factors. Primarily, we've been looking at drilling, so combi drills seem to be quite effective. These are quite common farm types of equipment. Typically, the most important thing here is to have good soil moisture and good soil to seed contact. So the combi drill seems to be able to do that quite well. We've also seen growers use precision planters, or more like a vegetable drill, which gives a good placement of the seed and can handle that small seed size well. So where available, they also provide quite a good option. Some consolidation, depending on soil type and moisture, is also quite good. So to try and get that inhibition and germination quite quickly. Because one of the big problems with these solanaceous species is that they are not native to the UK. They come from more subtropical climates, And so we need to really get them going as quickly as possible. Typically they're sown about May, June time, so moisture can be a bit of a problem. Heat not so much normally, although in the last few years we've had up and down successes with that. Another consideration is pre-emergence herbicides. So currently there aren't any products registered for use on these, but we've been trialing a number of products that could be used, and they are examining a registration process to enable them to be used on these crops. Because of that, kind of slow to establish, particularly in cool seasons, weeds can rapidly outcompete them. So we need to try and get the crops up and out of the ground as quickly as possible. We've also shown the importance of nutrition. So whether that be any residual nutrition in the soil from previous crops, or whether that requires an input of manures or synthetic nitrogen to try and get that biomass. Typically with decyst, the first decyst, Solanum cimbrifolium, also known as decyst prickly, it's in the region of about 700 grams of dry matter, fresh weight per square meter that's needed to get those good exudates. So it's about 7 tons to the hectare. And here you can see in the picture with my colleague Bill, the flowering crop there, that's a good sized biomass, but that needed some input of fertilizers. Alongside the agronomic side, we're also looking at the glasshouse efficacy testing, as I mentioned. So we've been working with Harper Adams here to look at the effects of the 3 species, the decyst prickly, the broadleaf and the podium, on how efficacious they are against the 2 species of potato cyst nematode, the Globodera pallida and Globodera rostokiensis. For decyst prickly, we're seeing in the region of about 70 to 80% efficacy. So when grown optimally and when given the best opportunity, they can have quite a high level of efficacy. But we're still looking through the data for the other 2 species because they might offer something different. We've held a variety of demonstration events and days across the country, which have been well attended by growers. These are primarily where these photographs have come from. And we hope to also have some videos coming out as well. So if you look out on our social media, there'll be some more in-depth videos with some stakeholder commentary on the project as well. So moving on from decyst, we're now going to look at potato light. This is a different project working with Grimmer, Pepsi and McCain alongside a stakeholder of universities and other growers. Here we're really looking at that elephant in the room of cultivation. How can we reduce the amount of soil that we are moving and the cultivation intensity? So I think, to me, this photograph really demonstrates the need and the kind of problem in many ways. Traditionally, we grow potatoes in this manner, lots of machinery, moving lots of soil to try and get a good seed bed and a good soil, good opportunity for the potato to grow and produce good tubers without blemishes, without stones and things like that. But as we are seeing a lot of growers turn to alternative sort of regenerative practices, this type of activity can be quite negatively impacting on wider rotations where growers have moved to minimum tillage or are using cover crops, trying to build soil organic matter, for example. So is there a way that we can either reduce the depth or the intensity of cultivation whilst also maintaining the good quality and yield that we see from this crop? Also, we have very little information as to how damaging, if at all, these practices are. They seem quite extreme, but actually are they as long-term as we think in terms of the impact that they are, or damage they are creating, or are they even damaging in the first place? And so we're also looking at soil health measures and greenhouse gas emissions associated with different cultivation regimes. So the project's aims are fourfold, really firstly to try and develop a novel lower intensity cultivator solution. So can we either reduce the depth or the amount of soil that we're moving in those cultivation stages? Could we go to something more like a single pass system or a minimum tillage type style for potato? And also, how do we deal with stones and how do we try and prevent them from getting into the crop and also through the supply chain? We want to quantify and benchmark the impact of these cultivation strategies. So both what growers currently do and also these novel lower intensity, lower depth or reduced pass systems that we're developing. So we're looking at examining soil health using a variety of metrics and greenhouse gas emissions. And this is really where the expertise of Harper Adams and Cranfield University come in and have been very helpful. And we've been monitoring these factors in the field this year in our first year of field trials. We also want to deliver practical solutions for growers. So we're working with a variety of growers across the UK to implement this. And we're doing field trials that are at scale rather than a small plot experiment, so that these are very practical long runs of potatoes that a grower would experience to try and get the most information out of that. Alongside that, we're going to develop an economic impact assessment. So are there any economic impacts that could be beneficial to a grower in terms of capital investment or running costs, alongside measuring those kind of environmental impacts. And then finally, trying to pull this all together into a cohesive story that we can tell to supply chains and to also consumers, and ultimately look at trying to move the industry towards practices that reduce the impact of cultivation in a variety of factors, as I've discussed. Just as a quick example, typically we're working with 4 growers who are spread across the UK. We've turned these as regenerative systems, so they use a variety of minimum tillage and cover crops elsewhere in their arable enterprises, and non-regenerative systems, which are using more traditional plough-based systems. We're overlaying trials across all of these sites, and these trials, the pilot trial is on the right-hand side, are at a field scale, using full-sized field equipment supplied by Grimmer and also the growers themselves. So finally, one of the kind of loosely projects that I wanted to talk about was just to also raise awareness of our crop storage and post-harvest solutions. This is a collaborative new research centre that we've set up in collaboration with ADAS, NRI, JHI and SAC and Shrug. And really this is to re-establish that strong network of post-harvest research capacity that we had in the UK. So prior to this project, there was the AHDB Sutton Bridge Research Facility, which ran for many years and was well respected by the industry. But following its recent closure, this left a bit of a gap in the UK's infrastructure. So CHAP, in coordination with these research partners, decided to set up a new network facility across the UK and invest in new capabilities to enable this crop research, storage and post-harvest solutions to continue going on. So we've received capital investment from UKRI and also from the Scottish Government and the partners themselves to establish a new network of storage solutions, whether they be much larger 6-ton storage facilities or smaller mobile 200-kilogram peapod facilities that can be moved into growers' stores. And really the point of these is to enable the UK community to continue to develop new solutions, whether that they be sprout suppressants, new storage regimes, not just in potato, but also in fresh produce and soft fruit. And also, can we better examine novel technologies that are coming through into stores, both for their management, for the detection of pests and diseases that might develop in stored produce, and also for that monitoring of sprouting and sprout suppressants. So looking to the future, just as a bit of a summary, Agritech really provides us with a lot of opportunities for the key challenges that I outlined. But one of the things I wanted to leave people with was the idea that Agritech is not just about shiny solutions, as I think we've heard about today in Bactobio's own project, where it's more of a biological based solution. So traditionally, we might see Agritech as just being sensors, data, AI, the big shiny new technologies. But to me, Agritech, it also means some more biological solutions, whether that be solutions like Bactobio developing through biopesticides, whether that be looking to integrate much more holistic solutions into our diverse farming systems, to redesign farming systems so that we can look at cross rotations, at cultivation regimes, IPM, those types of approaches, and also how technology can enable us to better manage the landscape and to take that bigger picture that might not always be possible. So look at how we could manage the landscape for environmental schemes, such as planting woodlands or pollination strips or certain environmental side kind of schemes. how it can look at that much bigger picture, enable farmers and individual farms to also connect things up in the landscape. And with that, I'll leave it and pass you back to my colleague Richard. Thank you very much.
Thanks very much, Alex. Very interesting. One thing I forgot to mention at the beginning of the introduction was the help that we had from ADAS and Alderley in particular in finding potato blight, particularly last year when, as we may remember, it was extremely dry and there wasn't very much potato late blight around, but we eventually found it in Aberystwyth. And this year was the complete opposite, where we had lots of rain and lots of blight. So we repeated the trials or the sampling in Aberystwyth again this year, again with the help of Aldwyn from ADAS. So thank you very much, Aldwyn. So if we go through the questions, starting the first one. Question from Catherine Lambourne. Do you think the soil microbes you're detecting when plants are affected to a lower severity are actually turning on the host's defenses of the potato plant? Or is it a different mechanism? Who wants to have a go at that? David, would you be best placed to make a start?
I could certainly make a start. I mean, there are examples of so-called elicitors or compounds which will stimulate defense mechanisms within the plant. But I wouldn't like to speculate on whether the actives that Mark and his team are going to develop have that mode of action or others. So I'll definitely pass that back to Mark.
So I definitely think that some of the microbes that we detect will be turning on host defenses in the potato plant. We're also interested in especially interested in the microbes that are producing compounds that will affect the pathogen itself. And so our screening assay at the moment doesn't look for any kind of elicitors of host defense and rather focuses just on the fungicidal compounds.
Very good. And Ryan Barry from AAB is asking a question about how do you mitigate for being unable to find a compound that will target a specific pathogen?
Yeah, so we have a diverse library of microbes. And so the work that Maisey explained with prioritizing those that have biosynthetic gene clusters for compound production is one way that we prioritize our library. In terms of a mitigation, our main mitigation is our library approach. So having a very diverse library of lots of different pathogens, lots of different microbes. And we also employ methods that upregulate production of compounds. So like Maisey said, some microbes might have 50 biosynthetic gene clusters, but they aren't necessarily all on. And so we have various methods for exploiting these gene clusters and getting them to produce lots of different compounds.
Thanks, Mark. And do Bactobio have plans to extrapolate to other oomycetes and fungi after the initial 18-month validation stage? Because we heard today that we're not talking about a fungus at all with potato blight.
Yeah.
So, do we need to think of a new term for a potato fungicide?
An oomyceticide, I think, is probably the word, but a bit of a mouthful. We are looking for compounds that target various different pathogens. So we currently have 4 bacterial pathogens. We have 1 fungus, 1 oomycete, 2 fungi and 1 oomycete. And at the moment, because of the transferability of our platform, we can target new pathogens. It's rather just about limitations on throughput. As you can imagine, if you've got screening campaigns against each of those pathogens, that kind of cascades down, and there's a significant amount of work in the compound isolation stage that needs to be done. We're always open to exploring new pathogens though, so if anyone is particularly interested, they can get in contact, and we can talk about ways in which we could look at screening against a new pathogen.
Thanks, Mark. And the next question is directed at you, David. Can you see the question?
I can, yes. So do you want me to go read it? Yeah, as someone's commenting about recent samples in Northern Ireland being of the 13A2 lineage. So should we recommend continuing use of the fungicide metalaxyl, I assume they're talking about there. I would recommend in all work is to look at the proportions and likelihood of sampling any particular lineage in this, the season immediately before and early in the season you want to apply the fungicides. So I mean, at Hutton we're offering a service through the Fight Against Blight campaign. We provide samples and we provide answers within a week. So early samples can be tested, find out what genotype there is, what are present in the crop, and then alter the program accordingly. So I would just, certainly it's, I would advocate care, but if we look at total proportions, then, I think you can evaluate risks based on the proportion and the chance of it being found.
Thanks, David. And a few questions from James Lee. When looking for potential biocides, how do you believe bacteria in the soil affect the ability of late blight to infect leaf tissue, which is somewhat related to the first question, Mark?
Yeah, exactly. So I think I'll take those three questions from James. So the first one is that Phytophthora, the pathogen, has a reproductive life cycle that is in the soil. And so whilst it does infect the plant on the leaves, it also lives and grows in the soil. And so we working on the assumption that the microbiome is affecting the Phytophthora throughout its life cycle. And so you can imagine that if there's a microbiome that is protecting against this pathogen, maybe killing it within that sample, then it's unable to infect the plant later in the season. James also asked whether we've identified any microbial actives yet, and if they're protective or curative, and how long do we believe they will be active for. We have identified active compounds here in this project. They're currently undergoing the plant trials, so we don't yet know whether they are protective or curative. What we have seen in the past is that our purified compounds are better at eradicating the disease than protecting against the disease. And we do believe that that is associated with the stability of those compounds. And so we haven't yet undergone appropriate formulations to say whether or not they're better at protecting or eradicating. And then finally, James asked whether we believe that these actives would have single site or multi-site activity. At the moment, we don't know. We will be doing mechanism of action studies for the compounds in order to find that out.
Very good. And a question from John Clarkson to Carrie or Maisey. How do you link the microbiome results, the 6 beneficial bacterial groups, to what you can actually isolate in culture for fungicide development, i.e. are they from the same groups? If so, do you pre-screen culture bacteria using 16S sequencing to identify these before whole genome sequencing?
Yeah, so I can answer that. So yes, we do pre-screen cultured bacteria using 16S, and then following that on the bacteria that we find are novel, then we'll do whole genome sequencing on them to elucidate the biosynthetic gene clusters, as Maisey went into more detail on. And for the first part of the question, the groups that we identified as being related to disease suppressive soils of different phylogenetic groups, so they're not closely related bacteria. And in order to try and culture them in the lab, we take a systematic and data-driven approach to build up information on what kinds of bacteria grow on what kinds of nutrients, so that we can isolate them in the lab.
Very good. And another question for David. Have you considered measures to tackle migration of the Maisey from other countries with some fungicide resistant genotypes into the UK or other regions worldwide, considering migration of different genotypes could potentially occur through the transportation of infected tubers in addition to sexual spores?
Yeah, this is a tricky one. Thanks for the question, Segun. Obviously, Phytophthora infestans can just simply blow across the channel, so that we can't do much about that really. So that's one pathway, but it's just being aware of it, and that hence the Euroblight and the Fight Against Blight programs is really just awareness and making people aware of the risks of that and the implications. In terms of seed, yes, I mean, there are restrictions on the infection of blight within the international seed trade. There are thresholds set. Has to be said, the threshold is fairly high, so it would still relate to several infected tubers per hectare of planted tubers. But I think I would say I advocate to growers growing the highest possible health tubers you can, and if you can get the tubers you need from a more local source, that's, you know, in principle going to be a better idea than getting them from a source that's further away. Movement of soil, that just relates, you know, in terms of the sexual stage which lives in the soil, just people need to be incredibly careful about not moving soil from one country to another, and just minimize the risks as best we can. But this is about awareness as well, so people are ready for a threat. If you know it's on the horizon and you know what it's going to bring, then being prepared for that is important. I don't know if I just could I just briefly follow on from the question about below ground and above ground that I responded to right at the beginning. Just a thought that, you know, suppressive soils, you know, we've heard of those in terms of take-all decline and things like that in cereals, but I'm not really aware of any suppressive soil type effects in blight, certainly not in Europe. I do wonder if it might be worth testing soils from further afield where there is actually a longer coevolution of pathogen and its host microbiome. You know, obviously, I wouldn't advocate moving soil from places where, perhaps in Central America or South America, where the pathogen and the host have been co-evolved for decades, you know, for millennia. But it's just a thought. You know, it's not a particularly natural system, Phytophthora infestans and potatoes, since it's only been around since 1845. So it's a bit interesting thought to look further afield in other projects, perhaps.
Good point. Right, an easy question for you, Mark. How long before these will be developed commercially available to growers?
Yeah, that's a very difficult one to answer. I would say that we are looking for natural products in our methods, and so these typically have a slightly faster route to market because the compounds are often kind of share a lot of the characteristics that you look for in a fungicide. So that's things like solubility, delivery to the pathogen, specificity for the pathogen itself, and also stability. However, we do anticipate that it will be several years before these are commercially available. We're looking at licensing these compounds out for further development. So we will take them to a certain stage and then hand them on to larger agrichem players to commercialize further. We have actually just been awarded another project to follow on from some of this research as well, which allow us to take the compounds further down the development phase.
Very good. Thank you, Mark. Question for Alex. Would the planting of decis plantlets in peat blocks be commercially viable? This would give a more consistent stand in the field and quicker crop establishment.
I think the short answer is probably not. So a lot of these trap crops are going in the wider arable rotation typically. So they are being done in place of a wheat or a cereal crop. So a grower there is wanting to do quite a quick action and also to try and minimize the costs. So they probably won't have access to plant raisers or to transplanting equipment. So being able to put this seed through conventional arable drills for cereals is probably the quickest and easiest and most cost-effective way of doing it. That being said, Nick Taylor, one of the growers on the project, who is more of a vegetable grower, has used that vegetable drill, so that sort of onion type drill, to precision drill them into beds. So that approach proves quite well. So if you do have a transplanter and you do have availability to access them, then that might be something that could work out, but I think it's probably unlikely. So in most instances, like I say, we're seeing combi drills, all those kind of precision drills, just to put the seed directly in, and hopefully with the improved kind of understanding of the agronomy, we should be able to improve that establishment without needing to raise them in a greenhouse and then transplant them out as well. Do you want me to follow on to the next question as well, Richard, about the...
Yes, please, Alex.
So in terms of the species, probably just give you a little bit more flavour. So the original DSIS product was the prickly, which is Solanum cisimbrifolium. That's the picture I used in that slide with great big spikes. So that's been on the market for some time, and we have some pretty good information about that. But it's just about some final tweaks, which I think we've made in the course of the 2 years. The second one is broadleaf, which I briefly touched upon, which is Solanum scabrum, which is also sometimes known as African nightshade. And I believe is used as more of a sort of spinach kind of edible leaf type in certain parts of Africa. There we know a little bit about it, but we're expanding our knowledge of it. That might offer some better characteristics as well over the traditional prickly. And then the third one that we've been looking at is the much newer species, which is Solanum chenopodioides, which is relatively unknown and relatively new in terms of PCN management. So we've greatly expanded our knowledge and understanding of that over the last 2 years to try and give, again, something different on the market. Those are the 3 species that we've been looking at as part of the project, but there are lots of other solanaceous species. How much they've been examined elsewhere, I'm not sure of, but those are the 3 main species we've been dealing with for the last 2 years.
Thanks, Alex. And an opportunity, Mark, for you to tell us how frustrated you are that CRD did not recognise a formal biopesticide category.
Yes, thank you, John Williams. It is frustrating. I think one thing that's particularly interesting is the differences worldwide in terms of the processes you have to go through to get these compounds to the commercialisation stage. And we are looking forward to some changes in the UK as it adapts to being a bit more of a standalone player in that area. And then I've got another question here. Shall I go on to that one, Richard?
Go on, yep.
So John Clarkson's asked if we thought about commercialising the bacteria themselves as biopesticides rather than secondary metabolite products that might fit better with the current direction of travel of crop protection products and may, or hopefully, accelerate registration. We are looking into live microbial solutions. One thing that is challenging for us is that these microbes don't grow very happily on their own. So our technology allows us to bring them into the lab, and they have quite specific conditions that allow them to thrive and produce these compounds. And so what you'll see is a lot of the live microbial solutions at the moment are kind of all mostly from bacillus strains rather than anything else. And so what we are looking at doing is taking the DNA that's making these compounds and putting them into a more stable strain that could potentially be a live microbial solution. But at the moment, we haven't been successful with that. What we have seen is that when we take the whole extracts, so all the compounds that are being produced from a specific strain, they often have very good protectant and eradicant activity. And so we hypothesized that there's something going on in the whole extract that allows compound stability and delivery better than the individual compound itself.
Thanks, Mark. There's a question which you may have already answered to some extent from Mike Abraham. What's the route to market for your discoveries, direct, or are you expecting to partner with an outcome manufacturer or even be bought out? Or what kind of timeline would you expect?
So at the moment, our plan is to partner with a large player to take these compounds further. We've had various communications with these players, but we're still figuring out exactly what those licensing deals would look like. And the second half of that question was how long we anticipate it to take. Is that right? Yeah. I think realistically, we're looking at 5 to 10 years before these compounds will be commercially available. We are trying to build a platform that does routinely deliver these compounds. And so whilst it seems like quite a while to wait from today, what we hope to do is to be consistently delivering new solutions across a range of diseases.
And a question from Radek. Do you have data on field trials with Bactobio isolated microbials controlling blight that you could share? I don't think we do we?
No, so this was a 18-month feasibility project where we went from soil microbe through to the plant trials. We will be taking these compounds further, and over the next couple of years, we'll start to be collecting some of that data. Yeah, a lot of that depends on funding and project progression and how those compounds look in the trials.
And another question from John Williams asking about the shelf life. What's it likely to be for novel biopesticides?
I don't know. I think as we take the compounds further and start thinking about their formulation and production at scale, I think that's when we'll be able to answer that. But right now, I'm unable to answer that.
Yeah, it's in the very early stages, and we don't have too much detail on stability, do we, of these compounds?
Yep.
And there's a question from Mohammed Hassan. He's working on bacterial endophyte mediated management of Phytophthora infestans of potato. He has a question about the limitations of bacterial microorganisms to overcome Phytophthora infestans. That one, Mark.
Yeah, sorry, I mean, one of the major limitations is the time it takes to identify the compounds that are active for bioactivity and then purify those and produce them at scale. I think what we see is that these bacteria, they don't grow well. That's why people haven't cultured them before. They then produce compounds that are all very similar to one another. And so isolating out the active compounds is hard work. And also them producing enough compound that is commercially viable is something that does take a lot of optimization.
I just chip in there as well, just to say, I mean, it is a challenging pathogen. It's known to be aggressive. It's very well adapted to attacking its host in a susceptible host. But Phytophthora infestans, and all Phytoptheras, they have vulnerable phases. They do have these motile zoospores which swim, and they're without a cell wall. So, that's a potential vulnerability of them. And I think we just have to work with their vulnerabilities and try and stop them infecting or stop them sporulating within hosts. But yeah, I mean, it's difficult, but I think we need to understand a bit more about the ecology of these things and how and not only for Phytophthora infestans, but for other Phytophtheras in natural ecosystems and in horticultural systems. There have been studies on composts, for example, and compost exudates and things which have an effect, whether that's directly on the pathogen or through eliciting resistances, we don't know. But also we need to consider combinations, as I kind of tried to point out in my talk, point out combinations of host resistance and an active of some form, see if we can tailor the optimal combinations of those. So it's worth thinking about that too in the long term.
Thanks, David. And there's a question just come in from Anne McPhail. Is there any work being done with Black's differentials? If anybody knows what that is, because I don't.
Okay, that's a great. Well, I can certainly answer that one. The Black's differentials are the R gene containing differential series. They have a collection of genes from 1 to 11. And yes, I mean, we do virulence. So what we do is for all those novel strains that we detect, we test the virulences of them, which means can they overcome those R genes in those differentials. So yes, that work is being done at Hutton routinely, but we accept and understand that the R gene differentials are not all the R genes which exist for potatoes. There are others which are being bred in through the Commonwealth Potato Collection, for example, and other sources. So we need to be aware of all those things.
Excellent. All right. Well, thank you very much, panelists, for your presentation and answering these questions. And thanks to all the participants for joining today. And yeah, if you've got any questions, any follow-up questions, then we'll probably send out something afterwards so you can have details of the presentations and of our contact addresses, so you can get in touch with us directly to ask any further questions that you have. And yeah, once again, thank you very much to everybody.